Dear ladies and gentlemen, dear guests, dear colleagues, for today's panel, it's going to be online, of course, and we are going to cover a very interesting subject about the COVID-19. And this subject has never been maybe, or in some circumstances, the problem is discussed by many ways, but we are looking the panel, or we are looking at the subject of the COVID-19 from a different perspective. So the panel is not going to be on the vaccine or the vaccine production, or it's not going to be on the new types of treatments, models, or it's not going to be how we are going to protect the people from the exposure of the COVID-19 virus. So it's a different subject. Our aim here is to give some emphasis, just to give some idea about how this agent, how this coronavirus has emerged, where it has burned, and how we can overcome this problem. And from the starting of it has almost we are approaching to 2 years after the starting of this coronavirus pandemics. And now, as we are going, or as we are approaching maybe the end of this coronavirus pandemics, then the questions were rising on what is the origin, where we got this virus. So it's important to find out this supply, the source of this virus. And I'm sure that when the coronavirus pandemics is going to be over at the end, and these questions will rise more and more, and the people are starting to find out the reason, the source of this coronavirus. So it has happened, as I said, 2 years, and lots of countries, lots of cities have started to take some precautions, like the partial lockdown or total lockdown, but it caused lots of problems, especially from the economical or financial and social perspectives. So in this panel, although most of the people presenters are medical doctors, but we are going to see the financial effects and the social effects of this pandemics, and what kind of reasons we are going to have in any kind of occasions where, well, what is the way or what is the root of the coronavirus contamination? If it is, let's say, a natural source, what would be the effect on the global earth? From the financial section, or from the social section, and from the perspectives of the international affairs. And for example, if it's going to be discovered that it's a real bioweapon, so what kind of things the world has expected in the future? So we would like to emphasize especially these points from our presenters, not from only the medical point of view, also from the other ways, what kind of social and financial or cultural, maybe for international organizations, how they are going to react against any kind of solution, any kind of results. If it is laboratory leak, for example, what would be the punishment of the Chinese government, and what kind of things you are going to see, especially after the 2025. And I'm sure that because it's a very huge event starting at the end of the 2019, and there are lots of how can I say, lots of side effects, and which will happen, especially which will cover till the end of this 2020s. So that's why we are going to see, and these panelists are going to explain from their perspectives, and therefore I wish success from now on to our presenters, and I'm going to moderate, and every presenter, every speaker has 20 minutes, and after all the talks are ended, we're going to take the questions from the guests, if there are. But I would like to every presenter to give too much attention to their timings, and therefore I'm going to moderate the panel, and I wish success. And I would like to give the floor to Professor Mehmet Er Yılmaz, He is also competent and professional on the disaster medicine. I'm sure that he has lots of things to tell on the COVID-19 disasters. And the floor is yours, Mr. Arimas.
Thank you very much, Mr. President. Hi, everybody, and I wish you a good panel for all of you. Please accept my congratulations for your presentation from now on. After my president's speak, I'll very shortly brief you. I try to brief you about these panel topics. I have prepared a small introduction speech. Please let me please give some seconds. The COVID-19, especially the COVID-19 outbreak, which appear the first as atypical pneumonia cases with unknown etiology in Wuhan, as you know. Wuhan city of China is actually in the last days of the 2019, according to official reports, and spread to first Iran, and then through entire European countries, including mainly Italy, UK, and United States, has emerged also in our country in the mid-March 2019, and been on the agenda in our routine lives with extending quarantine and isolation measures. Since the epidemic broke out, scientists have claimed many considerations and findings about the origin and involvement of the coronavirus and tried to find answers whether the agent is a kind of biological weapon manufactured in the laboratory. Hypothesis and the anti-hypothesis on whether the virus is nature-made as a result of natural selection or human-made still remain on the agenda of the world. Despite the bans indicated in international conventions, possibly of being a bioweapon or revealing the birthplace of the coronavirus will lead the type of our expected features. So this panel explains the concept of biowarfare, the exact place of the coronavirus in the context of component of the biodefense, and will evaluate the virus from the aspect of biological weapon. At the end of my introduction, I wish again you all for a successful presentation and good discussion. Thank you very much, Mr. President. Here you are.
Okay, now according to our panel program, our presenters are going to make their talks, and the first presenter is Professor Dr. İpek Mumcuoğlu. I think she's from the Ankara Oncology Hospital Medical Microbiology Laboratory.
Yes.
And I would like to say some words about her. And she was graduated from the School of Medicine from Izmir. She completed her professional degree in medical microbiology. And she has 72 publications and more than 500 citations in medical journals, many book sections, and co-author of 2 national guidelines on microbiology. She is now currently working in Ankara Oncology Hospital and in medical microbiology department. And her topic is going to be on the, maybe it's going to be an introduction for the whole topic. So she's going to talk on the biological warfare and an emerging threat in the near future from this perspective. Yeah, the floor is yours, Dr. İpek.
Thank you very much. And I would like to start off by thanking Organization Committee for their kind invitation. And as you all know, from the beginning of COVID-19 pandemic, it was the most asking questions. if COVID-19 is a biowarfare agent or not. Luckily, I don't have to answer these questions today. And I look forward Dr. Tuğba Dal's speech. And my job is easier than hers. Today, I will talk about biowarfare agents that have been used in history and that may be used in the future, and new agents that have the potential to pose a threat in the future. The definition of biological warfare is the use of infectious agents such as bacteria, virus, or fungi. The intention the intent to kill, harm, or incapacitate humans, animals, or plants as an act of war. These agents may be lethal or non-lethal, may be targeted against a single individual, a group of people, or even entire population, and they may be used by nation-states or by non-national groups. There are three definitions. They all use the same biological agents, but the target group are different. As I said before, biowarfare targeted the entire population. Bioterrorism is the intentional use of biological agents against a civilian population. Biocrime is the intentional use of biological agents against a specific individual. Biological warfare agents has been around since ancient time, but their applicability on the battlefield has been unpredictable, and they have blowback possibility and uncertain efficacy. But new powerful technologies could potentially be used to generate new biological weapons. So they may lead us to think biological warfare has a future, and the society must prepare for it. The use of biological agent is not a new concept, and history is filled with examples of their use. For example, 400 BC, Scythian archers infected their arrows by dipping them in decomposing bodies or blood mixed with manure. 300 BC, dead animals were used to contaminate the water sources by Persian, Greek, and Roman armies. 12th century, the bodies of deceased soldiers were used to poison wells during the Battle of Tortona. In 14th century AD, during the siege of Kaffa, the attacking Tatar forces hurled plague-infected corpses into the city. 18th century AD, during the French and Indian War, British forces gave blankets that had been used by smallpox victims to the Native Americans. During World War I, the German army developed anthrax, cholera, and the wheat fungus specifically to be used as biological weapons. During World War II, Japanese forces operated a secret biological warfare. They exposed more than 3,000 victims to plague, anthrax, syphilis, and other agents. During the Vietnam War, Viet Cong guerrillas used needle-sharp fungi sticks dipped in feces to cause severe infections after an enemy soldier had been injured with them. In 1979, an accidental release of anthrax from a weapons facility in Sverdlovsk, Russia, killed at least 66 people. You can find some more examples in the history. In 1972, Biological Weapons Convention banned the development, production, acquisition, transfer, stockpiling, and use of biological weapons. And these biological weapons would be considered as war crimes. The Convention's restrictions applied only to signatory nations and are irrelevant to terrorist organizations or independent terrorists. So, since the 1980s, terrorist organizations have only become user on the biological agents. In 1984, 751 people were intentionally infected with salmonella. In 1994, a Japanese cult attempted an aerosolized release of anthrax from the tops of buildings in Tokyo. In 2001, I guess this one is the most famous one. Anthrax was delivered by mail to US media and some government offices, which resulted in 5 deaths. There were only 5 deaths, but the public panic was huge. And there are 1,200 infectious agents for human beings, but only few of them has the potential to ideal candidates to be a biological agents. The ideal candidates are easy to acquire, process, and use. Small amounts of them enough to kill or incapacitate people. They are easy to hide, difficult to detect or protect. They are invisible, odorless, tasteless, and it can spread silently. Sorry. The CDC categorized bioterrorism agents in a 3 category, from most dangerous to potentially dangerous. You can see the whole list here, but we are going to take a closer look. And in category A, they are high priority agents. They are easy to disseminate or transmit from person to person. They have high mortality rates, potential for major public health impact. They cause public panic and social disruption, and they require special action for public health preparedness. We need to talk about these all pathogens. Anthrax is normally a non-contagious disease caused by Bacillus anthracis. It's a spore-forming bacteria, and it's endemic disease. It is also endemic in Turkey, and it causes typical skin infection. Treatment is easy, but inhalation of spore is fatal, more than 90%. When discovered early, anthrax can be cured with antibiotics, and vaccine is also available. Smallpox is a biological weapon, very dangerous because of its highly contagious nature. It's an airborne virus and has a high mortality rate, and it killed about 500 million people from 1880s to 1980s. Smallpox was eradicated from the world in the 1970s thanks by vaccination, but currently people are unprotected in the event of a new outbreak. Tularemia is very infectious. 10 to 50 organisms can cause disease. It is commonly found in nature. Transmission occurs from contact with animals' fur, inhalation, insect bites, or ingestion of contaminated water. And clinical presentation also make a different way. Tularemia has a very low fatality rate if treated. Inhaling an infectious aerosol causes severe respiratory illness and maybe death. If Francisella tularensis was to be used as a weapon, the bacteria would likely be made airborne for exposure by inhalation. Botulinum toxin is the deadliest toxin known to man. Lethal dose for a human is less than 2 micrograms. Clinical syndromes ranging from food poisoning, wound infection to infant botulism. Botulism causes death by respiratory failure and paralysis. Furthermore, the toxin is readily available worldwide due to its cosmetic application in injections, but doses are very low. Plague is the disease caused by Yersinia pestis. Rodents are the normal host of plague. Disease has 2 clinical presentations. The most common type of plague affects the lymph nodes, and it's called bubonic plague. It can be severe, but there is no human-to-human transmission. The deadliest and most rapid form of plague occurs when it reaches the lungs. It can be transmitted person to person via droplets in the air. It is considered a threat due to its ease of culture. The weaponized threat comes mainly in the form of pneumonic plague. Viral hemorrhagic fevers, you can remember Ebola virus outbreaks. It caused very large public panic. The viral hemorrhagic fevers agents there are two families of them, Filoviridae, Aranaviridae, and most famous one is Ebola virus. Ebola virus disease has caused high fatality rates. That is due to multiple organ failure and hypovolemic shock. The arenavirus have a lower fatality rate, but are more widely distributed in Africa and South America. In category B, there are some known pathogens. Brucellosis, you can see Clostridium perfringens, you can see Salmonella in this category. In this category, there are second highest priority agents. They moderately easy to disseminate, moderate morbidity rates, and low mortality rates. In the category C, there are third highest priority agents. Includes emerging pathogens that could be engineered for mass dissemination because of their availability, easy to produce and disseminate. There are high morbidity and mortality rates, and they have potential for major public health impact. In this category, there are multi-drug resistance mycobacterium tuberculosis, Nipah virus, hantavirus, chikungunya virus, and some strains of influenza virus and yellow fever. And how are biological agents delivered? They can deliver it through the air by aerosol sprays, used in explosive, put into food or water, absorbed through or injected into the skin. And what are the common epidemiological clues that may signal biological attack? From most specific to least specific, an abundance of cases of unexplained disease and deaths, unusual presentation of the disease, unusual, rare, genetically engineered strain of an agent, unusual geographic or seasonal distribution, unusual trends of death and or illness in animal population. This could be the earliest signal. When we talk about the future of biological warfare, there are 5 major existential threats to humanity. Previously, infectious diseases were not considered a cause of extinction. However, some environmental microorganisms could potentially pose a much greater threat to humanity than the known pathogenic microbes. For example, fungi have a high bioweapon potential, and certain amphibian and bat population extended by fungal diseases. Potential future threats encompass all microbes that inhabit the planet. For example, as you know, normally avirulent Aspergillus spp can be highly pathogenic for immunocompromised hosts. And at least 335 new infectious diseases have been described since 1940. Most of them are zoonoses, and examples HIV, coronavirus, Ebola virus, Legionella. They are all zoonoses. It is almost a certainty that humanity will continue to confront new microbial threats and that many of these agents, such as HIV, flavivirus, possess a significant weapon potential. And genetic warfare, I think we need to talk about it more than anything else. Theoretically, biotechnology could be used to design novel types of biological warfare agents, super agents. Such an agent could demonstrate how to render a vaccine ineffective, confer resistance to therapeutically useful antibiotics or antiviral agents. That means you can't treat Bacillus anthracis anymore. They can enhance the virulence of the pathogen or render a non-pathogen to virulent, increase the transmissibility of a pathogen, alter the host range of a pathogen, enable the evasion of diagnostic tools, and enable the weaponization of a biological agent or toxin. And there is a new method which called clustered, regularly interspaced, short palindromic, and which called CRISPR-Cas9 systems This system has emerged as a promising technique for gene editing. In this technique, a DNA sequence is cut off and replaced with a new sequence or code that codes for a particular protein or characteristic, which could potentially show up in the desired organism in a very short time, maybe in weeks or days. CRISPR-Cas9 is cheap and widely available. This method can cause serious issues and potential danger if used by people with the wrong intentions. As a last slide, I would like to say emerging threat in the near future. What could they be? All biological agents on the CDC bioterrorism agent disease list had been used in the history, and they have the potential reuse in the future. And we can see new infectious diseases as a zoonosis. And genetic warfare, especially synthetic biology, could be used to design new agents. And as I told you before, some innocent fungi extinct, some amphibian and environmental microbes could be very dangerous for all people. And I am usually a positive person, but it's really difficult to stay positive when you are talking about biowarfare agent, because it is reality. Potential future threats may cover all microbes that inhabit the planet and maybe more. The only thing that we can do to be prepared. Thank you for listening.
Okay, thank you, Dr. Ipex. So from your presentation, I think the situation is hopeless. So we should wait, or we should expect some more dangerous microbiological agents in the future. Or, again, according to your presentation, so we can say lots of microbes or lots of like the zoonotic agents or others are going to emerge in the future. So it's important if we are going to be prepared, or if you are going to overcome all these coronavirus pandemics, this is going to be a good lesson learned or good preparation for our next threats, especially coming from the new microorganisms. So thanks, first of all, for this very good introduction for the biological warfare agents. And our next speaker is from the USA, and Mr. Dr. Rashid Chotani. Is that the right pronunciation? I'm not sure. Chotani
yes.
or Kotani? Right, Chotani, yeah. And he is my good fellow, and I think more than 10 years we are in a good relationship with him. And if we say something about the bio of the Dr. Rashid Chotani, he's the recipient of the 2016 NATO Scientific Award for Counterterrorism, and he has spent over 20 years providing biodefense and infectious diseases like the influenza, coronavirus, Zika virus, Ebola, and Crimean Congo hemorrhagic fever, and that virus. And he's also a professional on the public health and emergency management to expertise the public agencies, private industries, and academia and non-profit organization. He is a very good recognized expert, and I'm sure for that in medical countermeasures, development and biosecurity, healthcare management, health surveillance, and diagnosis of human and zoonotic diseases. He has applied to this expertise to groundbreaking research and development and advanced development of programs for naturally occurring non-made or genetically modified biological and chemical agents. So currently, Dr. Chotani holds, as far as I know, the College of the Professional Studies, and now he is also the professor of epidemiology at the University of Nebraska Medical Center. Now, also he has some studies at the George Washington University with a focus on the public health, emergency, and crisis communication. So, Chotani, I think you're a very good expert on the chemical and biological defense. So I'm sure that you are going to tell lots of good things or bad things, I'm not sure, because how these coronavirus pandemics is visible from the perspective of the United States affairs, and I'm sure that you have lots of new things that we are wondering from your presentation. The floor is yours, Dr. Chotani, please.
Thank you so much, and thank you for inviting me to this meeting and to the panel. I hope that you can hear me properly and you can see my slide on the screen.
No problem. Yeah.
Good. So it is a very important topic, and today I will talk a little bit about some of those possibilities in terms of, what is this virus, where it came from. But what we want to do first is to understand what is COVID, what it's doing, the lessons that we've learned in the US, so that we can apply those in terms of lessons that have been learned to other countries, which have also learned a tremendous amount of lessons. But there have been some successes and there have been some failures in terms of responding to this virus. So as I start all my discussions, we still don't know much about Sars Coronavirus 2. And I also say that at this particular point, I don't call myself an expert, and I don't think anybody is a total expert in COVID or SARS Coronavirus 2 specifically, because the entire book has not yet been written. We keep learning about new things every single day, and then we question ourselves as to where this virus has come from. Having said that, we all know that coronavirus has been in existence for a very long time. We've had human coronavirus, which is OC43, which is called common cold, as well as 229E, which is also a common cold. So those two viruses have been circulating and causing some minor issues across the globe for a very, very long time. Then, of course, we had the SARS coronavirus outbreak, severe acute respiratory syndrome, in 2002 and 2003. Subsequently, we saw in 2012 MERS coronavirus, the Middle Eastern respiratory syndrome, come about, and that has stayed and has persisted at a very low level and still is causing disease in the Middle East. The specific coronavirus responsible for the disease called COVID19 obviously is SARS Coronavirus 2, which we started observing last year, or in fact in China, perhaps a year before in the month of December. Today, as we look at the world, we see that this disease has caused a tremendous amount of havoc. And there's a particular reason that I have this slide on the screen, is to make sure that everybody from an epidemiological perspective is also aware of the cyclical process or cyclical cycle that this virus has. And if you dig deep down into it, it seems like that this virus, which we thought would be essentially a seasonal problem, would be coming during the fall and winter, does not hold the same pattern as other viruses do, but it comes and goes every few months. And we just hope that, right now, as the numbers are going down, as we go into the fall and the winter months, in most of the world, except a few which are on the other side of the equator, that this the surge will not happen again. But so far, about 5 million people have died, which is a very conservative number, in my opinion, because a lot of countries have not reported the deaths. And once this disease is over, if we get out of this thing, then we will find out exactly how many people were affected and died. Obviously, the economic damage, the social, psychological damage this virus has caused is also tremendous. As we look at how things are today, you will see that looking at the world, you saw 3 major waves. But when you look at and break it down by the different regions of the world and take a look at the data from per capita perspective, you still see the waxing and waning of this pandemic. And as you can see right now in the United States, which is the red line, or Canada and United States, the numbers per capita are going down. But you see that the light blue line, which represents Europe, the numbers are going up. And if you go back in time, you can see that every time the numbers in Europe go up, the rest of the world, the numbers also go up. You know, they're close by, they're close to each other. So you can look at one region of the world and see the numbers climbing, and then obviously viruses do not respect boundaries, so other regions also get affected. So my fear is that in the next few months, even in the United States, the numbers will start going up again. When we look at the hotspots in the world, you can see the higher, the darker the red is, the more cases are being observed. And as you can see, that in Europe, parts of Europe are extremely affected. And if we zoom into this entire map, you can see that, of course, Turkey is becoming a hotbed, and the surrounding countries are also very, very much affected. And pretty soon, you will see in the next few months that the virus is going to surge again in a lot of these other countries, which are neighboring countries to Turkey and Bulgaria and the rest of the European peninsula. So just to give you a quick idea as to what the US response was to COVID-19, which in many experts' opinion, in my opinion, was not the optimum response. But what we did was it's something incredible. We created a process which was Operation Warp Speed, through which we accelerated the production of vaccines, as well as therapeutics, as well as diagnostics. So the time period required for developing therapeutics and diagnostics is not as as long as when you talk about vaccines, but this process worked, and we've developed some new techniques and new processes through which vaccines can now be developed, which is a very positive thing. For the production prioritization, we went through a lot of different processes and made sure that the distribution and the administration of the vaccines was being taken care of in a very systematic way. And it's a very good model, provided that people are willing to take the vaccine. We had incredible distribution processes in terms of coverage, in terms of uptake and following, and various methodologies and processes through which we monitored how many people were getting the vaccine, you know, what was the response, and how the guidance was coming out. There was a lot of confusion in terms of even the CDC guidance that was coming out for the vaccines or the therapeutics or for the diagnostics, which caused some serious issues and problems. The journey, this slide I call the COVID vaccine journey. So how, you know, when it's produced, and then how does it go about in this entire nation, which is a very, very large nation in terms of geography as well as population. How does it go from the production line all the way when the patient receives? And there are multiple steps involved in it. And the reason for this slide is to make sure that when the slides are shared with the participants, they can use it and perhaps utilize it if need be for the purposes that they are, for the work that they are doing right now in public health. The medical research during this pandemic was unbelievable, right? So we were able to develop vaccines, diagnostics, and therapeutics during a raging pandemic at a very rapid rate, and the entire world participated in it, which was unbelievable. Unprecedented number of scientific publications or papers were, published in peer-reviewed journals. So there was so much work that was being done that if you look at the history of the world, it's unprecedented amount of work that happened during this pandemic, and it's still going on. But the unfortunate part is that we still have not been able to control the virus. So what is in control? The virus is in control. We know that there's science behind wearing masks. We have shown through data that wearing a mask helps in a major way. We've shown that vaccinations decrease hospitalizations. We've shown that monoclonal antibody treatments will decrease hospitalization and severity of disease. All of this data is so obvious and so clear, and apart from the rest of the world, in the United States, as far as vaccines are concerned, we have no problem and no deficiencies. We have more vaccine that we need at this particular point. We have monoclonal antibodies. Masks are also available, but unfortunately, a large population, a large segment of the population, neither is using a mask or is getting vaccinated or is using monoclonal antibodies. And that is a very serious problem that we face, and that's a belief system problem in terms of people believing in something and not believing in what science and data is showing them. So there's so many lessons, and I just want to quickly go through some of the critical lessons that I think are important for us to understand. The first thing that we all understand, and I can assure you that even in a bio-threat kind of a scenario, that you will, and the rest of the folks on the call would appreciate that you've got to act very, very, very fast. Part of each country's experience with COVID has been extremely different. And why it has been different is because it all depended upon initially where the virus landed and when. What was the area, which city, which geographic area, and what was the timing of it. And then a structural factor in terms of the population age played a very important role in how things happen. South Korea was one of the first countries to face the COVID outbreak. The country controlled it with mass testing, mandatory contact tracing, having already gamed out what it would do in this scenario because they'd been practicing this thing. And the graph on the side of the chart demonstrates what was going on during the early phases of this pandemic in Korea in terms of testing and what was happening in the United States. So the United States started out very, very late, and it took them at least 2 months to even get to the level of per capita testing that Korea was doing. By that time, it was perhaps too late. Islands like New Zealand and Taiwan enjoyed structural advantages in terms of the age factor, as well as the island itself, which was isolated, but they still had virus introduced in the country multiple times during last year. They moved quickly to snuff out the clusters and prevent the virus from escaping containment. So that is a very critical thing, escaping containment, and they were able to do that in the United States and a lot of other countries. We were not able to do that. South Korea passed public health reform acts after MERS outbreaks when they experienced the worst outbreak in the Middle East. I mean, this was early in going. They invested a tremendous amount of money in hospital infection control units and biotechnology sector, which I believe every country and every nation needs to do. They ran periodic pandemic war games, these exercises to walk through outbreaks and scenarios and hypothetical situations as to how to deal with it. Within weeks of detecting the first cluster, South Korea was running most of the tests, as I showed in the graph earlier, in the world. And they quickly established contact tracing so that they could control and contain the virus. Both South Korea and Vietnam acted before the full threat of coronavirus was appreciated by the rest of the world. So acting fast is extremely critical. Then being flexible and creative. So you got to be flexible in terms of what you do, in terms of when a pandemic like what we are facing right now occurs. South Korea established the biopharm sector to develop diagnostic tests for COVID before anybody else. They moved things so quickly, it was unbelievable. They created telephone booth testing technology and tested every single person that they could. The United States, on the other hand, centralized the test development to CDC, discouraging other scientists from developing their own. Other countries were not doing the same. The problem was that in the US, the tests that were developed by CDC were faulty. So this was a major, delay in the response. Vietnam started to close its border to China at the beginning of January and mid-March, early in the game, and sealed itself entirely from foreign travel. Over time, Vietnam seemed to have been proven right. Its highest single day count of cases has not been more than 110, which is unbelievable compared to the rest of the world. In Germany, one of the cities in Germany, based upon one small hospital study, suggested that they were, going to mandate masking, and initially they did. And the rest of the country was masked, and that helped very much during the first phase of the pandemic. Later on, they obviously changed, and people were tired of wearing the mask, etc. And that again caused a serious problem in Germany. So the countries that acted quickly found it easier to adapt as the pandemic wore on. South Korea and Senegal, even in Africa, have had second and third waves of the infection, but they were prepared because they had reacted very early during the first cycle of this pandemic, and they were able to crush that wave during the first pandemic. So now the second and the third, they were much, much more prepared and better in terms of what they were doing. In US, however, the virus quickly escaped containment. We were not able to contain the virus at all because we were not accepting the problem that was here, and we lost the window to suppress it. So suppression, which is lesson 3, is everything. If you can suppress the virus, and the countries that were able to do so were very successful. COVID-19 has been widespread ever since, with the US never recording fewer than 20,000 cases per day since April 2020. Even today, we have close to about 100,000 cases every single day in the United States. So suppression is critical. Every country with drawn-out pandemic is struggling or has struggled. Germany initially succeeded, but later on failed because people were not ready to mask. In the US, though most people did wear masks and practice social distancing at first, those habits became more and more politically polarized over the years. And it's a political problem. In April, Democrats and Republicans were almost equally likely to say that coronavirus was a major threat in the US, and that is this year. By August this year, they were saying that, hey, 85% of the Democrats were saying it's a major threat, 46% of the Republicans agreed. So that became a huge problem, a political divide, and it became a political weapon rather than a public health issue. So when so there was some data that you can see on the side of the graph. It seems like Asians still most likely to wear face masks due to COVID, and the numbers are clear. Vietnam, China, Italy, Japan, India, US comes at 50%, France, after that, Germany now at 20%, where it used to be a very, very large percent of the population was wearing masks at the initial phase, and the UK, 16%. So slowly, a lot of these countries are stopping wearing masks, and that is also helping spread the disease. Lesson number 4, politics should not trump public health and science. So public health and science and the data should be forefront and be used rather than political rhetoric to deal with this disease. A raging outbreak exacerbated raging outbreaks as political divisions. So the Republican and Democrats, the right wing and the left wing were competing for what they thought was the right thing, and they used it as a political way of winning elections. And it's not just in the US that this happened. It happened in other countries where actually governments were toppled because of COVID. So COVID also played a major role in changing the political landscape, and then politics was used and abused and COVID was used for political gain or political loss. And that is what not should have happened. And as you can very well imagine, if you have a brain tumor, you go to a neurosurgeon. If you have an infectious disease outbreak, you should have the experts who are leading this cause, either infectious disease or infectious disease epidemiologists. And all of a sudden, we had so many infectious disease epidemiologists around the world, and nobody was using them. They are the ones who are the experts in this field, and they should have been upfront. But then experts came around, and there were so many experts that we can't even count them anymore. So U.S. is a massive politically divided country with complex federal systems of the government. Has structural problems which could end up weakening the resistance to new diseases, no matter what containment protocols are in place, because we have lack of universal health, we have pervasive chronic disease problems, we have a huge problem with just obesity, diabetes, hypertension, heart disease. We have decentralized health system. So those are the factors that are going to cause a problem in the US and wherever in the world those systems exist. The current pandemic is not over, and the cost of inaction is too high. The biggest mistake we can make is to think that we've defeated COVID. We have not. And a lot of politicians all the time come out and say, for political gain, that now we have defeated COVID. There should be an urgency about preparing for the next outbreak, whether it's a naturally occurring outbreak, whether it's a lab accidents, or whether it's a man-made pathogen that is being dispersed in the world. So we need to be prepared. So let's talk a little bit about fact and fake, right? So everybody talks about, the conspiracy theories. So I just wanted to make sure that I touch that because the next speaker is going to talk a lot more about it. But the conspiracy theories usually start to appear on social media immediately after the first news comes out of any crazy or infectious disease thing or anything that is detrimental to humanity. But in this case, it was COVID. The big question was, is the virus a hoax? A lot of people in the United States, around the world, still think that this is a hoax. There's no virus. Is it a bioweapon designed in a lab? So that is another big question that is out there. These conspiracy theories either blame a group for having some involvement in manufacturing the virus or controlling public opinion about it. As biodefense experts, we have to look at the three potential scenarios. One is the natural occurring and evolving virus. The second one is lab leak, and it's something being produced in a lab, somebody working on a virus, making it virulent, and then by accident, it gets released from the lab, or somebody gets infected and then goes outside in the community, infects somebody else. Or an intentional spread. When we look at this data that you are seeing on the side of the screen, there was a recent survey done, and the question was asked about the conspiracy theory regarding this virus. And as you can see, unusual, right? Unusual answers to that question. People were saying that, hey, the pharmaceutical companies are doing something because they need to make money and they want to come up with a miracle cure, and whoever comes up with a miracle cure is going to make a lot of money. There's a new world order, a deep state that is causing that problem. The Republicans in the United States were saying the Democratic Party it's a hoax, it's a lie, there's nothing happening. Then people saying that it is something that came out of the Wuhan lab, a bioweapon. People said, about 2.5% people said that it was Bill Gates who was behind this thing. A lot of people said it was the 5G technology that the phones had. Then it just went on, and including Dr. Fauci was blamed by the population. About 1% of the survey takers said it was Dr. Fauci who's done this thing. And then, of course, bat soup was part of that equation also. So conspiracy theories and how things disperse in terms of the media and people talk about can be of different natures. And as you can see, at least in the United States, you can clearly see, this was a study that was done by Cornell University. You can see that Wuhan and bioweapon labs, created at the Wuhan lab was not at the top of the chart. It was there, but not at the top of the chart. And Bill Gates was about at the same % level as that particular theory. The virus is in control. Irrespective of how this virus spreads, we need to find a way to defeat it. History will decide what caused this pandemic, because we will not know for a very long time exactly what happened. Currently, it appears that it is a naturally occurring pandemic, and the virus is mutating, evolving due to external pressures. As you know, all of you know that viruses also want to survive, and once we start developing ammunition against it, they will start evolving, they will start changing, and this virus is acting in a very peculiar way. Our primary responsibility as individuals who are in healthcare, who are here to save lives, is to be proactive, not reactive. Find solutions to this problem, save lives. That is the most important thing. And then base all our decisions on science and data. This is the last slide that I always show people in terms of what needs to be done, what is a personal responsibility and what's a shared responsibility. This is called a Swiss cheese model. And you know, you talk about different things that you can do for your population, masking, avoid touching your face, fast and sensitive testing and tracing, government messaging and transparency, financial support, vaccines, now monoclonal antibodies or other therapeutics that are out there. Those are different layers. If you put all those layers, you will decrease the number of transmission, you will decrease the number of disease. And once we can bring it down to a certain level where our hospitals are not overwhelmed, the deaths are not the way they are today, even today with the numbers going down, we might be able to control it in a much better way. And I thank you for your time and your patience. And at the end of the discussion, whatever questions there are, I'd be more than happy to take. Thank you, sir.
Okay, thank you, Dr. Rashid. So it's a very explanative presentation that I indeed have learned lots of many new things. So I mean, from your presentation, so we have lots of lessons learned, and it seems that we are going to have more and more lessons learned in the future. So, but it's important, especially you talk about some conspiracy theories, but sometimes we have to remember that some realities can be born from some conspiracies. So, and we are also very sure that it's visible. It's going to be visible after maybe the coronavirus is over, both in the United States or in the rest of the world. So I'm sure that lots of questions can be asked at the end of the panel about your presentation. I'm sure that also that our guests are preparing for the questions. Maybe I'm sure that also you're going to be with us at the end of the panel, so you're ready for the answering the questions, right? Okay, so the next speaker, our third speaker is Professor Tuba Dal, and she is the professor of the microbiology, and she has also a PhD and doctorate degree on the public health. She is working, and she's graduated from the Istanbul University School of Medicine, and she completed her professional degree also in the same school on the medical microbiology. And she has more than 100 publications in medical journals, many book sections. She is the editor of the book titled, especially in the last year, The Current Laboratory Medicine, and is also the co-editor of the book titled Coronavirus Disease 2019 and the Turkish Perspective. She is a co-editor of this book, and I'm sure that she has lots of references in her talk. And now she's working in the lab as a laboratory supervisor in Ankara Oncology Hospital, and on the medical microbiology department, and she's also the lecturer in this branch in Yıldırım-Beyazıt University School of Medicine. So the floor is yours, Dr. Tuba, and her talk is going to be on the SARS-CoV-2. It's a bioterror agent with the question mark, or has another source with another question mark. So I'm sure that she is going to clarify these question marks in her talk. Yes, the floor is yours, please, Dr. Tuba.
Thank you, Dr. Kenar. I'm sharing my slides. I think, okay, is there a problem? Okay. Yes, I'm starting. Sorry. Yes. Today I will talk about coronavirus is a bioterror agent, or it has another source. We know we have confused about these questions, and now I reviewed the literature and I try to talk about the source, the origin of coronavirus. So the aim of this presentation is reviewing the origin of SARS-CoV-2. We know that since the first reports of the COVID-19 in Wuhan, China, there has been a discussion on the origin of SARS-CoV-2. So for understanding these arguments, firstly, I want to show you the classification of coronaviruses, because for understanding this, we have to learn this knowledge. Coronavirus family, there are 4 genera. You can see here alpha, beta, and gamma genera. And some of the human coronaviruses are classified in alpha coronavirus, and some of the human coronavirus can be classified in beta coronaviruses. And MERS-CoV, SARS-CoV, and today's topic SARS-CoV-2 were classified in beta coronavirus, you can see. And coronaviruses can cause mild respiratory system infections, and these are human coronaviruses, and they can be classified in alpha and beta coronaviruses. And the significant human disease causing coronaviruses, we know SARS-CoV, MERS-CoV, and SARS-CoV-2. And if we look at the history of coronaviruses, the earliest reports of the coronavirus infection was acute respiratory infection in domesticated chicken, emerged in North America in 1920s. And then the first name, it was called infectious bronchitis virus. So it is an animal virus. And then the other coronaviruses were found in 1940s. The first one, brain disease in mouse, and the other one, mouse hepatitis virus. And also then after this, in 1960s, in United Kingdom and United States simultaneously, human coronaviruses were found, and virologist David Tyrrell and his colleagues proved coronaviruses are responsible for the common cold. And until now, in microbiology or in infection disease, we know that these coronaviruses, human coronaviruses, can cause infections, mild symptoms in the human, and there were common also in the world and in our country. But then coronavirus outbreaks were seen in the world. The first one, SARS-CoV. For severe acute respiratory syndrome coronavirus, SARS-CoV is coming from this, appeared in Chinese Guangdong province again in 2002, and this outbreak had over 8,000 infections worldwide, and the fatality rate was 10%. And then SARS-CoV, this virus, crossed over the human from the bats, Himalayan palm civets, and raccoon dogs. And the other coronavirus, MERS-CoV, the Middle East Respiratory Syndrome coronavirus, and caused large-scale coronavirus outbreak in 2012 and transferred to human from the infected dromedary camels. And according to genetic analysis of different viruses, it is believed that it may have originated in bats and later transmitted to camels, and then in short-chain infections, it transmitted to the human. MERS-CoV infected fewer people than SARS-CoV, but the mortality of MERS-CoV was very, very high. You can see the mortality was 36%. And the other our topic, SARS-CoV-2. In December 2019, it was appeared in Wuhan, in central China, and quickly showed human-to-human transmission, and it was called a pandemic by the World Health Organization, and the name of the disease was COVID-19. And you can see here, according to the classification, some of the COVID viruses coronavirus story, highly pathogenic, and the others are low pathogenic coronaviruses. And for understanding if it is a bioterror agent or not, maybe this is a microbiology topic, but you have to understand the structure of coronaviruses because they are spherical particles, and here you can see they have some spike glycoproteins on the surface, and they are enclosed in an envelope. They have a lipid layer, and this envelope is encoded by the membrane, M protein, you can see, and envelope protein. And also for us today, the spike protein is very, very important. And you know, it is the target of the vaccine also in many vaccine, and also it is responsible for the infectivity. So spikes are very important. And spikes consist of S proteins, and these proteins are needed for interaction with the host cells. You can see this is the spike protein and this is the host cell, and by the S proteins we will see an interaction. And here you can see the S protein is composed of S1 and S2 subunits. And please focus on S1 subunit. S1 proteins are the most variable components, and they are responsible for host cell specificity. And here you can see the receptor binding domain. I think you heard many things about receptor binding domain, mutations, etc. They are responsible for recognizing the protein receptors, such as angiotensin converting enzyme in COVID-19. But in the other coronaviruses also, it can bind to another protein receptors. And the other part of the other subunit of S protein, S2 subunit, and it encodes a spike in the envelope. And for understanding, you have to learn the spike receptor binding. Firstly, the initial binding of angiotensin converting enzyme receptor, you can see, and then spike have to be proteolytically activated, and then the spike is cleaved at the S1 and S2 junction. And then S1 dissociates from the S2, and we can see the fusion of the S2 and the host cell. And it is important for the infectivity. And I said the receptor binding domain is the variable part of the genome, and it is very important for the infectivity. Yes, what are the notable genomic features of SARS-CoV-2 from the other SARS-like viruses? Firstly, if we understand the SARS-CoV-2, firstly, the first one, SARS-CoV-2 has differences in receptor binding domain. Yes, receptor binding domain there are in other coronaviruses, but SARS-CoV has some differences. And also, SARS-CoV-2 has both polybasic cleavage sites and 3 adjacent predicted all-linked glycans in the structure. Firstly, I mentioned about SARS-CoV-2 has differences in receptor binding domain. Why? Normally, 6 amino acids in receptor binding domain are critical for the angiotensin converting enzyme receptor binding and host range in SARS-CoV-like viruses. But the differences of the SARS-CoV-2, 5 of these residues differ between the other SARS-CoV-like viruses. And so, because of the differences in the receptor binding domain, SARS-CoV-2 has higher affinity to binding angiotensin converting enzyme in humans, ferrets, cats, and other species with receptor homology. And also, I mentioned about SARS-CoV-2 has both polybasic cleavage site and 3 adjacent predicted all-linked glycans. Yes, SARS-CoV-2 has a polybasic cleavage site at the junction of S1 and S2, and we cannot see this site in the other lineage B beta coronaviruses, and SARS-CoV is classified in this group, and this is different from SARS-CoV. But in some lineage A human coronaviruses, we can see this polybasic cleavage site. And you can, what is the importance of this polybasic cleavage site? There are some studies, laboratory studies about this. For example, there are some differences, monobasic or polybasic. And if there is a polybasic cleavage site, the spike protein harbors a multibasic S1, S2 site, and cleavage at the S1, S2 site is essential for the spike-driven viral entry into the lungs. So for the angiotensin converting enzyme binding, it is very, very important. So it is important for the viral infectivity and the host range. And what is the role of polybasic cleavage site on the S1 and S2 junction? There are some animal models about it. For example, you know, there are many studies previously about SARS-CoV, and if normally there is no this furin site, or the other name polybasic cleavage site. But if in a laboratory we insert a furin site to the SARS-CoV, we saw that S1, S2 junction, if there is a furin cleavage site, it enhances the cell and cell fusion. Also, there are some studies about avian influenza virus, and if there is a polybasic cleavage site, and in highly dense chicken population, some of the avian influenza viruses can gain this junction, polybasic cleavage site, and can transfer to highly pathogenic forms. So it is very important, very significant for us. And for understanding, I said this is the structure of the coronavirus SARS-CoV-2, and this is the S1 subunit, S2 subunit, and this is S1, S2 junction, and here you can see the polybasic cleavage sites. And there is the other difference here. Here you can see the all linked glycan residues. It is important for understanding because in SARS-CoV-2 there is a difference because there is proline here, and also there is an inserted polybasic furin cleavage site, and the sequence is PRRA, and there are some considerings that it can be responsible for the addition of 3 different all-linked glycans. And so we can understand there are some differences in SARS-CoV-2. What are the main role of these all-linked glycans? Why all the virus produce these all-linked glycans? Because all-linked glycans are the sugars, and they can add it to serine and threonine proteins. And what are the importance of these all-linked glycans? They could create a mucin-like domain that like a shields on the SARS-CoV spike protein, and also the other several viruses, for example, Ebola, can have all-linked glycans. But I said polybasic cleavage sites and different all-linked glycans, 3 different all-linked glycans can present in SARS-CoV-2. So after understood the structure of SARS-CoV-2, we have to discuss the scenarios of SARS-CoV-2 origin. And there are 2 scenarios that explain the origin of SARS-CoV-2. The first one, natural selection in an animal host before zoonotic transfer. The second one, natural selection in human following zoonotic transfer. And the third one, today's topic, the laboratory origin. Is it possible? The first one, the natural selection in an animal host before zoonotic transfer. What is the meaning of this? This scenario suggests that there is a bat coronavirus, and this bat coronavirus gained polybasic region in bats, and there are many bats in the world, and naturally selected in the bats, and then passed to pangolins, and in the pangolins, receptor binding domain recombinated and optimized for the binding to human angiotensin converting enzyme receptor and then passed to human. Yes, why we think it can be? Because the early cases of the COVID-19 were in human market, and it is possible that an animal source was present at this location. And the other evidences, Rhinopulus affinitis bat virus. This is a bat virus. This is a SARS-CoV-2, SARS-CoV-like bat virus. And now in microbiology, we can analyze the genetic similarities of the viruses, and many studies there are about it. And we saw that the overall SARS-CoV-2, there is a 96% genetic similarity. But there is a differences in Rhinopulus affinitis bat virus because the spikes of the bat virus may not bind to human angiotensin converting enzyme 2 because there are some divergences in the receptor binding domain. So, but after many, these researchers studied and screened some animals can be the intermediate host. And we saw that the pangolin coronaviruses receptor binding domains have all 6 key residues, and there is an amino acid similarity segment. And this shows that the spike protein may be optimized for binding to human-like angiotensin converting enzyme 2 is the result of natural selection in pangolins. And so what we thought about this, we know that neither bat nor pangolin coronaviruses have polybasic cleavage until now. There is no animal coronavirus as a progenitor of SARS-CoV-2. Maybe in some bat coronaviruses, mutations, insertions, deletions occurred near the S1 and S2 junction, and then a natural evolution occurred and polybasic cleavage site arise, and in a high population density of bats, natural selection occurred. This is the first scenario. And why we think bats might provide a poor genetic diversity? There are some studies that the bats might provide a poor genetic diversity for the original SARS-CoV-2. And also, there are some studies that many mammals, some primates, rodents, and carnivores could serve as an intermediate host of SARS-CoV-2. And in a study showed that pangolins has a high capacity for binding angiotensin converting enzyme 2. So the pangolins as possible intermediate hosts. And also, SARS-CoV-2, the idea is SARS-CoV-2 may be a genome, a chimera genome with the bat and pangolin coronaviruses. This is chimera in Greek mythology. And the horizontal gene transfer and recombination analysis studies show that SARS-CoV-2 could be a chimera virus. It can be a recombinant virus with bat virus and pangolin coronaviruses. Yes, this was the first scenario. And the second scenario, natural selection in humans. This scenario suggests that there is a SARS-CoV-2 progenitor virus. We didn't know now, we didn't detect. And in the human, it acquired genomic features for adaptation. It, for example, acquired polybasic region, receptor binding domain, etc. And it naturally selected in the human. This is the second scenario. And current data point to emergence of the virus in late November 2019 based on retrospective studies. This scenario presents a period of unrecognized transmission time in the human. In the human, virus acquired the polybasic cleavage site in this unrecognized transmission. We don't know this period now. Human MERS-CoV infection is a result of repeated jump of the virus from dromedary camels to human and adaptation to human. There are some evidences about this, and so it can be the natural selection in the human, it is possible. The third one, today's topic, laboratory origin. Yes, claims. Some people suggested that it can be a leaking virus during laboratory studies by mistake. And the CEO of the Institute for Pure and Applied Knowledge made an online statement that SARS-CoV-2 was most likely constructed by a laboratory recombination. And also before these SARS-CoV-2 came to the Turkey, we have a discussion by ambassador of China, and in this period they were afraid and we don't know what will happen in the world. And the reasons for thinking that SARS-CoV-2 has a laboratory origin. Why we can think this virus is a laboratory origin? Because the resource involving passage of bad SARS-CoV-like coronaviruses in cell culture or animal models continuing for many years in biosafety level 2 laboratories, not 3, 2 laboratories generally. And then after this pandemic, the CDC recommended biosafety level 3 laboratories for cell cultures. And the other one, you can see the reasons for thinking that SARS-CoV-2 has a laboratory origin. There is here, you can see a publication in 2004 during the first outbreak of the SARS, 2 accidental releases from the Beijing laboratory. And in the, I have been in China before, but I looked at the map and you can see this is Wuhan region and this is the Beijing region. And there is some arguments that SARS-CoV released from these laboratories. And the reason of thinking that SARS-CoV-2 has a laboratory origin, the other evidence, SARS-CoV studies, not SARS-CoV-2, SARS-CoV studies observed mutations in the virus for the adaptation to cell culture. So we think that, yes, it can cause adaptation to cell culture. So maybe SARS-CoV-like viruses change, adapted to the human, but these viruses are produced in the laboratory. It can be. And the other one, Sirotkin et al. And colleagues showed that SARS-CoV-2 acquired spatial adaptation to human cells after serial passes in cell culture. So we can think maybe it can be a laboratory origin. And the other one, the reasons for thinking that SARS-CoV-2 may be a bioterrorist agents. SARS-CoV-2, there is a study and it was published in Molecular Biological's Portrait journal. And SARS-CoV-2 sequence is identical to bat coronavirus. And we know receptor binding is identical to pangolin coronavirus. And the natural recombination requires that the virus from bat and pangolin infected in the same cell, in the same organism, simultaneously. But the researchers think that it is improbable event because the density of the pangolin is very low and bat coronaviruses doesn't have significant affinity for the pangolin angiotensin converting enzyme 2. So they thought that this is a laboratory produced virus.
Dr. Tuba, can you sum up because we are a little bit...
Yes, I'm finishing. And the reasons for thinking that SARS-CoV-2 has not a lab origin, there are some studies about this because it has all linked glycans, and to producing all linked glycans is impossible in the cell culture because we need immune system for this. And there is a recent study. It showed that SARS-CoV-2 didn't contain a peculiar restriction site or other markers that suggested a manipulation in the virus. And the conclusions. Horseshoe bats seem to be a natural reservoir, while pangolins could be an intermediate host of SARS-CoV-2. However, the intermediate the source has not been confirmed until now, and there is no prior isolation of progenitor virus with a genetic similarity with SARS-CoV-2. Also, unnatural origin of the SARS-CoV-2 are strongly based on strict scientific evidence. There are some studies that this virus is unrelated to known artificial laboratory coronaviruses. But, however, it is currently impossible to prove and disprove the other theories of its origin of this virus, because yesterday I talked with my friend Ibrahim. I think he's listening us, and he said that it is possible a genetic manipulation without leaving a trace in these viruses. So what we will do, monitoring the variation of SARS-CoV-2 in genome, and we have to screen the animal viruses, and we need retrospective genetic analysis of SARS-CoV-like coronaviruses, and also the retrospective analysis of the humans. Serological studies are necessary for us. So I want to say that maybe coronavirus is not a bioweapon. We don't know now, but bioterrorism is a real future threat. We have to know. And this is my colors from Istanbul. And I hope to see you in face-to-face Congress in Turkey. Thank you very much, Dr. Kenar.
Okay, thank you, Dr. Tuba. And it is very good scientific explanation about the roots of the COVID-19. Maybe we are going to make a summary. So according to your presentation, so it is not a bioweapon, according to your presentation, but you are also leaving a gap. You're not just closing the definitions. So there might be maybe in the future some reasons that can conclude us that it's a bioweapon or a laboratory leakage. But I mean, from your perspective, from your presentation, you have some very good documented scientific publications. So these publications are significantly showing that it is just a natural, but you are also telling that there is an intermediate reservoir, that the bats, the horseshoe bats are okay, but we are the reservoir after the horse bat contamination, it is still unclear. So maybe the scientific world is going to enter another age to find out this intermediate reservoir before the human contamination. Okay, thanks for this very explanative. I'm sure that there are lots of questions can be asked at the end of this panel. So our last speaker is not a medical, but it's a very I'm sure that she is going to have a very explanative presentation, especially from the non-medical side of the coronavirus pandemics, and what we are going to see, especially in the economic or international area, especially after the coronavirus pandemics is over. So, Associate Professor Dr. Emete Gözügüzelli, and she completed her doctorate in 2017 at the Gurney American University, and his thesis was on international law of the sea in the field of the maritime delimitation, law in the Eastern Mediterranean. So she continued to serve as the advisor to the presidency at the Turkish Grand National Assembly between 2006 and 2008. She also participated in training on the security of the Eastern Mediterranean, and she gave a briefing to the members. And also she became a working partner with the Oxford University on asymmetric cultural relations in Cyprus, and worked as a lecturer at the universities in Turkish Republic of Northern Cyprus. And she worked as the head of the Law of the Sea Center at the Bahçeşehir Cyprus University. And she is also a member of the advisory board of centers such as Koç University Maritime Forum, and a member of the Ankara University Law Faculty of Maritime Law Research Center. And also she has many scientific board memberships. She also gave, I know her very well, she's one of my good friends for more than 5 years. And she gave many interviews on local and international TV channels and newspapers about the law of the sea and the Cyprus issue. She has also publications and books in many magazines and newspapers. And in addition, also, Emete was awarded the Peace Ambassador Award on April 29 in the year of 2018. So the floor is going to be yours, but I'm going to announce your topic, your title, Dr. Emete, and she is going to talk on the especially international law and pandemic era, and a canon law for change is also. It's a very interesting topic, and I would like to explain the topic especially in the interrelationship. Yes, please, Dr. Emete, the floor is yours.
Thank you very much, President, Professor Dr. Levent Kenar. This is really actually the historical time we will discuss the COVID-19 and its effects for the world in the case of especially the interstate relations, and in the case of the related to the international law. And first of all, before starting to giving my presentation, I would like to saying thanks to all of yours for valuable informations, and thank you also Kenar for invitation to me. Really, I am honored to be here, and thank you for all the participation again for their valuable informations, and thank you for the participants who is today with us. Yes, yes. Before starting, I would like to ask the first question: is COVID-19 a biological weapon? We know that we are discussing the topics in many fields, and it is really important topic. And we hear many thoughts about that, and in sometimes maybe some disinformations, maybe we can hear.
Dr. Emete, sorry,
So,
please click the slideshow button.
bottom?
Please click to slideshow button. Right
Yeah.
corner. Right corner.
Corner, yes.
Bottom
Is
of the
that
right
okay?
slides. Bottom of the right to magnify your slides. Right. Bottom right.
Bottom right.
Yes.
Where this one? Yes.
Kum saatini hocam. Kum saatini tıklayın hocam. Sağdaki, aşağıdaki. Aşağı. Yanındaki hocam, evet. Kum saat.
Şu mu
Hayır,
hocam?
ama yanında, hayır. Biraz daha sola. Evet
Dur.
hocam, orayı. Thank you so much.
Very much. Yes, of course, I'm not interested in conspiracy theory, but I would like to give you some information for that. We are talking about a viable war, and we are talking about the international law, and there is now such a title among global problems that we all need to think about it. And really, this is a serious issue. And the reflexes of relations between states in this crisis period are quite thought-provoking. And although we can't count COVID-19 as a biological warfare at the moment, but there are many issues that we need to think about. On the basis of the UN, of course, you know that the all states should attach importance to cooperation and solidarity. And now we are talking the survival of the people, and so in that case, we should ask that has the type of wars changed now, or where is the international law that all concerns the all states in the crisis? So before starting to discussion, I prepared some table of contents which I would like to look at the circles of biological warfare, and I know that it has explained in many times during the conference. And the effects of the COVID-19 on these interstate relations are important, and the international legal framework of biological warfare also my topic. And you know that the biological weapons are not new, and we have seen that it was used in various countries around the world. And İpek Hoca explained the many steps during that time, so I will not give many informations for that, but we have lived the same things during the history, and you will see in 13th century, in 17th centuries, many programs, many events actually happened related to the diseases, viruses. So in response to these detrimental impact which caused by the use of biological weapons in World War I, many countries signed the 1925 Geneva Protocol and a treaty prohibiting the use of chemical and biological weapons in international armed conflict. However, during the World War
Excuse
II,
me, Dr. Emeta, we
Yes.
cannot see your face, maybe because of the light problem. Is it possible to recover this problem? Because, yeah, in order to see the face.
I know, but I think it is not easy to actually.
Okay, I think some technical issue. So
Yes.
please go on. Yeah.
Thank
Okay.
you very much.
Thank you.
Yes, and then, however, during the World War II, and you know, the Japan violated the treaty by engaging in large and secretive biological weapons program and using them against the Allied forces in China. So at the end, we have faced the Soviet Union and the United States and the allies followed soon after the developed biological warfare production programs. So as a result, the first multilateral disarmament treaty banning the development, production, and stockpiling of biological weapons entered into force in 1975. And moreover, the Biological Weapons Convention has created a global norm. No country considers biological weapons to be legitimate means of national defense or any as such have a right to these weapons even as a means of deterrence. However, gaps in the convention to the treaty remains questionable. So we have faced many problems over the terrorism and their actions in the war. So we have lived some events in the case of the 2001 American attacks and anything else. So COVID-19 affect interstate relations really it's an important thing because the international system analysis is always concerned with the structure of the system and the interactions between their units and the implications for peace at war or cooperation and conflict of existence different types of the states. So the COVID-19 pandemic has affected international relations, and one group argued that COVID-19 caused diplomatic tensions, and as a result, the United Nations Security Council resolution demanding a global ceasefire, and also international relations is questioned on its topics, rethinking of existing approaches to international relations on the public health diplomacy in IR, the politics of crisis in IR, or war politics in IR. So other groups focused on the pandemic is unlikely to lead to significant changes in the international system. So diplomatic relations have been also affected due to tensions around trade and transports of medicine, diagnostic tests, hospital equipment for coronavirus diseases. And leaders of some countries have accused other countries for not containing the disease effectively and resulting in the uncontrolled spread of the virus. So in the case of the developing countries, insufficiency to cope COVID-19, we have seen many cases, and developed countries' restrictions overflights, diplomatic relations, freedom of movements, and the restrictions in public and private life affect all relations of the individuals and also the state of affairs during that crisis period. For example, a new world order design on diplomatic relations, we can argue and we can say that, of course, we are in the new world order designing on the diplomatic relations, because as you know that during that crisis, the countries gave priority to themselves and their health insurance was the first. For example, you can see Trump's steps here, and France also accused the United States of disrupting face mask deliveries by buying at the last minute, or there's some other events which happened from Canada. You know, the minister, public safety minister and transportation minister to investigate allegation that medical supplies originally intended for Canada were diverted to the United States. Some events you have seen that happened, and the EU also tried to make some steps, and many Schengen area countries closed their borders to stem the spread of the virus. But the realities on state affairs, for example, Italian Prime Minister commented that the whole European project risks losing its region data in the eyes of our own citizens. And especially in March, Germany banned the export of personal protective equipment, and in France also restricted exports of medical equipment, and drawing criticism from EU officials who called the solidarity. So also we have seen the division between the Northern and the Southern European member states during that process. And we have seen also the oil war. However, when Russia refused to reduce oil production, Saudi Arabia triggered an oil price war in March 2020. And this economic conflict resulted in a sharp drop of oil price over the spring of 2020, with the price becoming negative. And also Iran's regime's steps against the United States happened. And also cyber war. We have seen the cyber war steps and some attacks during that process. For example, the United Kingdom's National Cyber Security Center, together with the Canadian Communications Security Establishment and the United States Department for Homeland Security, Cyber Security Infrastructure Security Agency, and the United States National Security Agency in July 20, 2000, jointly alleged that Russian state-backed hackers have been trying to steal COVID-19 treatment and the vaccine research from academic and pharmaceutical institutions in other countries. But Russia, denied these events. At the end, we have seen that in 2020, United Nations Secretary General Antonio Guterres issued an appeal for global ceasefire as part of the United Nations response to the COVID-19 pandemic. At the end, we have seen that the 5 sectors have been affected during the crisis and the political affairs. For example, we have seen the insecurity era and the realistic approaches over the anarchical world has gained more importance, and the state as an actor also it's seen as an important actor. And in economic, in societal, and in environmental issue, we can see many events which affects the world during that process. So it is not related to political issues. It's not related to only the public health. It is related to all the environment of the world, which affect, in many fields, in digital field, in energy, in diplomatic, in many fields. So in that case, can ask that where were the universal values? Of course, will there be actual world war? We can ask. So we are of course already in the third world war, because this war marks a new world order, and there have been many transformations. We are in the digital world. This information has biological warfare risks have increased. Among these risks is an unstable and unpredictable future in the world relations. It has been seen, and the reason for this, this is the macrobiological risks, and we need to take it very seriously. So at the end, the world will progress not only with economic crisis, but also in many areas, including the health crisis, separate warfare methods, or other operations. And there are those who claim that the smallpox virus poses a threat to the future. Of course, whatever the name, COVID-19 has shown us that we are in a very weak world system. So states did not seek cooperation, but to ensure their own conflicts. Diplomatic shows were made during that process. So half the world is in serious crisis. We cannot ignore them. And our global problems, of course, are increasing. And to cope the global problems, the United Nations try to find some solutions to that, but it is not enough because the system of the UN actually is in the questionable. So this field is a very ahead, as it is really directly and rapidly affects human life. But we must prepare our, our work for the future with all the risks. And increasing nationalism under the pressure of the immigration, and we have seen many immigration events at the seas, and many inhuman activities happened, for example, by the some states, especially Greece, that some steps against these refugees. So we have seen many movements, we have seen many actions during that period. So of course, until now, we have seen many types of the virus. And at the end, we can say that, of course, diseases are also affected in the 21st century. So are we ready to another steps for the future to protect our world? Actually, it's not enough for it's not enough if you think some steps in the international arena. It's not enough if you will talk about the international law or the international relations, because still the world is not aware of the biological warfare. Of course, they prepared some convention during this 1970s period, but at the end, we should discuss and we should question what happened now. And the bioterrorism, it's a new program, I think, and most of the people don't aware that issue. But at the attacks of the biological weapons, for example, by the terrorist organization, which they target the civilian population, it is very important thing. But we haven't any steps to prevent any bioterrorism in the world. We should discuss it, because are we ready for the legal frameworks? We should ask ourselves, because the biological terrorism is a really important topic. And the universal definition, if you look at that universal definition of biological weapons, of course, the microorganisms such as viruses, bacteria, fungi, or other toxins that are deliberately produced and released to cause disease and death in humans and animals or plants. So United Nations expands this definition to say that they usually consist of two parts: an armed agent and a delivery mechanism. So biological weapons can be used for many reasons, including for military purposes, political assassinations, contamination of agriculture and livestock to cause food shortages, and economic instability, and epidemic that arouses fear and cause among the population. So UN, of course, tried to find some steps. For example, in September 2002, International Legal Framework Against Biological Terrorism events prepared, and most of the countries participate here, but it is not enough. Also, international IMO also discussing the issue, especially the safety of maritime navigation are also necessary to prevent acts of biological terrorism at sea. You know that. Because the shipping is a global industry and relevant to both landlocked and the coastal countries. Therefore, any country may have nationals who are victims or offenders of a terrorist act at sea. So, same legal framework is necessary to criminalize acts of biological terrorism for sea, at least for the international aviation protection. For example, 2010 Beijing Convention actually has taken for the biological terrorism. So what will be next? However, of course, the fact remains that whatever the origin of the outbreak, whether natural, accidental, or deliberate by state or non-state actors, societies need to be better prepared to prevent and mitigate or face the effects of epidemics and pandemics. At the end, biological warfare has been a serious concern for military operation and national security. And although the Sars virus and its variants are not normally seen as a deliberate threat, it is claimed that there are clandestine biological weapons programs and terrorist groups seeking such programs, and they take advantage of such natural outbreaks. Of course, at the point where the world has come, we see that the biological weapons have started to be discussed. So one of the most unexpected consequences of the epidemic has been actually to show how weak the foundations of international cooperation and global governance and international solidarity. And dimensions of the crisis have grown even more due to the lack of the adequate communication and the lack of the supranational coordination mechanism, even in crises that concern humanity at the global level. So we have seen the realistic theory was the agenda, which the main actor in domestic and foreign policy is the state, really confirmed once again, because the ineffectiveness of the international institutions, questions of liberal theory arguments was based on the international law and the relationship of the international institutions and other things. So we have learned many things actually in this process. So the issue of the biological attacks, it is seen that the biological weapons should be considered as a necessity in the development of the deterrent defense strategies. So that's why we should create the laboratories for examining, more laboratories for examining these events. And of course, the future, I'm not pessimistic, but the future still unpredictable process for the world. So of course, when we look at the international law-based dimensions of the issue in the face of all situations, we are faced with a new era and new types of war. Thank you very much.
Okay, Dr. Emete, thank you. Sorry for this. Very different opinion, because you talk about the international glance over this COVID-19 pandemics, and some very interesting sentences you talk on that topic. So I'm sure that this is the fourth and the last speaker. I'm sure that we have some questions, and so from the guests, so the floor is open to the questions, and the speakers are ready for answers. If there are questions, please, or any contributions.
Actually, I have a question to Tuğba Dal. Dr.
Yes,
Tuğba
please.
Dal.
Yes,
Thank
please.
you. I would like to know, in one of your scenarios, you said that the virus cannot be a lab virus. It was a scenario because of glycan addition. But as far as I know that some drug transformation procedures, glycans can be added onto liposomes. So maybe it can be adaptable to the cell cultures, isn't it? What do you think about this?
Also, before previously, I mentioned that all linked glycans can be added in laboratory conditions. And this was the other arguments of the other publishers. And they thought that we cannot add. But you are right. For example, in avian influenza viruses, for example, we can see these added all linked glycans. But there is a topic, there's an issue about it that for binding all linked glycans, we need to encounter with immune system of the human or an intermediate host, we don't know. And they thought that we cannot add in laboratory condition. But I think you think it can be in laboratory condition. Yes, it is possible. So it can be.
I was thinking about that. It's possible because of the liposome additions. Do you know how to? I don't know if you can have idea about this or not, because the drug delivery systems are generally composed of these kind of additions. That's why actually I think about it.
I'm not a biochemistry stuff, but in the studies, the virus, some of the virus can gain these all linked glycans during some passages in cell culture. But yes. I didn't see, maybe it is possible for your exam, but I didn't see any publication about the virus added all linked glycans or different all linked glycans in laboratory condition. I didn't see any publication about this. And the all linked glycans, yes, may be added, and there are three differences between the others. So a different all linked glycans produced and then added the virus.
Yes,
can be. We cannot say not, but it is difficult. I can say only.
Because as far as I know, in drug delivery systems, they are doing this kind of additions and just transform to the liposomes.
Yes.
Thank you.
Possible.
Okay. Thank you. Any other question? Okay. I have one question to Rashid, Dr. Rashid. Are you here?
Yes, I'm here.
Okay. I was wonder, for example, in the next couple of years, when the COVID-19 is going to be discovered or found out that it's a bioweapon, especially man-made by especially the Chinese scientists. So do you know or did you come up with any kind of conditions that if there is a situation like that occurs in the future, what would be the reaction of the United States government in that circumstance? Is there any kind of maybe the war can be expected, or any kind of financial crisis is expected, or any punishment, an international punishment can also be accounted for that reason? So do you think, or do you hear anything like that from your surrounding?
So yeah, there's lots of rambling. A lot of people talk about it, but in all honesty, we really don't know, first of all, if it is a bioweapon, was it intentional, was it an accidental, or as it looks like right now, based upon all the studies and research that has come out, there's a naturally occurring virus, and it has just become, it has evolved, and we are facing a natural enemy. Now, if something like this would occur, or if something like this was real, it would be a huge, huge, huge problem. We don't know what will happen then, because obviously there will be repercussions if something like that was inflicted. And we've had so many deaths around the world, and especially the United States has had so many deaths, and the burden of the disease has affected the economy in a very, very bad way, not just in the US but globally. So obviously, if this was something that was intentional and were discovered, there would be, the world would not be the same as it is today, unfortunately.
Okay. Okay, I see. So we have lots of things to see in the future, according to all the presentations. So, but our environment, are looking for some, for some result, and they are going to ask me, or they are going to ask you, is that a bioweapon or not? That's the question, and that needs to be answered. Or so, what's your proposal to me? I mean, what am I going to answer to that question?
So at this particular point, if somebody would ask me that question, I would say current information and the studies that we have looked at and the difficulty of making this into a bioweapon, we feel very confident that this is a disease that came out of the zoonotic population and jumped the species and came into humans. And now it has become an extremely virulent strain. And it's not the first time in the history of this world that we've had a pandemic of this kind. So this has happened over the course of the history of this world. And this is another chapter in the history of the world that is being written right now. And that is a major pandemic. And we'll have to see as we move forward if there's anything else that comes out that kind of assures us that this was not a natural phenomena, and it was a man-made element, or it was a lab leak. But we don't know as yet with any confidence. We cannot say that it was either the two bad scenarios that we talked about.
Okay, thank you. And I also any more questions from the guests, from the listeners?
There's a question from the participants.
Yes, please.
To Dr. İpek. Is there any method that can detect biological agents from the environment?
Yes, there are some. I know some electrochemical sensors for all category A agents. I mean plague, for tularemia, for anthrax. There are some electrochemical devices. I'm not quite sure if they are available commercially, but I know they exist.
There is another question is to Dr. İpek. What to do for biodefense against the biological agents?
Okay, first we need to understand of their biology and what are the interactions between our immune system and these biological agents. And we need to find drugs, new drugs and vaccine. And of course, we need some research laboratory to find out how can we defend this biological agent. I remember this. I can say that. Thank you.
Okay, so if there is no more question, I can just sum up. First of all, I would like to thanks to the speakers, thanks to the guests, thanks to the listeners for their very great contribution, for taking the time. And also, I would like to thank to my co-moderator, Dr. Ariel Mals, and for those talks of Dr. İpek, Dr. Tuba, Dr. Rashid, and Dr. Emete for their very nice talks. And they are very clarifying talks. And I'm sure that we are going to remember this panel in the future. And maybe next time, maybe next year, if we do the same panel again, we can have more and more clarified points on this, the root of this COVID-19. And I'm sure that we're going to prepare a kind of the meeting report, and it's going to be published in our website. And at the end, I could say best wishes and thanks for your efforts, for your preparations, and thanks for the time for the others. And I would like to say goodbye to everybody. And I'm going to terminate this panel, and hope you have a very good week, and hope you very good news in healthy and safe, and to be removed from the COVID-19 exposure. Yes, thank you for all. Bye.